So it's really helpful if you can think about this in terms of evolution. So the key word here, like I said, is evolution. Yep. So with evolution, if you have a population that has phenotypic variation, so all the members of a population have a certain trait that's not the same amongst all of them, but that this trait is heritable in that it's passed from parent to offspring, well then a selectional pressure placed on this population should cause the population to adapt and evolve in response to that selectional pressure. So in terms of Darwin's finches, Darwin realized when he went on his voyage to the Galapagos Islands that the population of finches on a certain island was somewhat determined by the food source available. So if on an island there was only hard nuts, Darwin astutely observed that, well, in response to the food source, in response to the selectional pressure, the phenotypic trait exhibited by the finches was going to be long beaks that were capable, long and sharp beaks capable of busting open those nuts. So I mean, it works on birds. It also works on RNA, as it turns out. So in our case, the selectional pressure, okay, so let me back up. In our case, the phenotypic trait is the shape of the molecule. And our phenotypic selection, it's not going to be nuts or food source. Instead, what it's going to be is going to be ability to bind a certain protein. So with that in mind, this is the selectional scheme. So I start with my random RNA library. So 10 to the 13th molecules. And the way we get 10 to the 13th molecules is we start with a synthetic DNA that has a 40 nucleotide long random region. So if you guys can do, I'm sure someone knows that 4 to the 40th is a lot larger number than 10 to the 13th. 4 to the 40th is such a large number that it'd be too many sequences. I think you'd need like 2 liters of DNA in order to cover that number of sequences. So instead we use something less. We use 10 to the 13th. And because DNA is easier to synthesize artificially than RNA, we start with the DNA library. But then we transcribe it to RNA. And then we incubate the RNA with my protein. So the protein of interest in this case is lysyl tRNA synthetase, so LysRS for short. So we incubate the random RNA pool with the protein, and then we have to separate those RNAs that bind to the protein from those that don't. And the way we do this is we pass it through a nitrocellulose filter membrane. So nitrocellulose is a polymer that has a negative charge. And as you guys know, like charges repel, whereas opposites attract. So RNAs, nucleic acids, polynucleic acids have a phosphate sugar backbone. And these phosphates possess a negative charge. So these negatively charged molecules won't be able to stick to the nitrocellulose filter. Whereas protein, my protein, LysRS, is going to have a partial positive charge. So that allows it to be retained on the filter. And therefore, if there's any RNA that's also stuck to that protein, that will also be stuck to the filter. So that allows us to select out only those RNA molecules that bind to the protein, while all others flow through the filter. So after we've separated the pool, we need to regenerate the size of it. So to go back to Darwin's finches, it's not going to happen overnight. It's not going to happen in one generation, where the population evolves to be adapted to the food source. It takes many generations, 10, 20, 30, 40, 50, before you can actually start to see some sort of selectional bias. Well, it's the same idea with RNA. It's not going to take us 40 rounds. It usually takes us about 10 to 15. But the point is we need to do the selection procedure again. And in order to do that, we need to regenerate the size of the pool. So at this step, I usually only get back maybe 1% of the RNA I used to start with. So I need to re-amplify it. And the way I do that is I elute the RNA off the filter, and I use a technique known as RT-PCR. So RT, reverse transcription. So I take the RNA and I transcribe it back to DNA. And I have to transcribe it back to DNA so that I can use polymerase chain reaction to amplify the pool. So once that's done, I can start it all over again. So it's kind of a simple, not simple, but a repetitive process. I've kind of been on it for 2 years, and I've had a lot of, I've had to stop and start over again. It's kind of been a long and tedious process. But right now I think I'm onto something. So I think we're actually we're making some headway. So after 7 rounds, we can see a noticeable increase in the affinity of the pool. So as you can see, at round 0, my protein, or my pool bound to the protein with an affinity of 400 nanomolar. So what that means is it means it takes 400 nanomolar of protein to bind half of my RNA pool. Now after 7 rounds of selection, my KD is down to, it's in the 80 to the 90 range. So that means it requires 5-fold less protein to bind the same amount of RNA. So the RNA binds the protein that much tighter. So I'm not done yet. I still have another 4 or 5 rounds of selection to go before I can move on to the next step. And the next step is going to be to set the pool off, sequence the individual molecules, test the individual molecules for how they interact with the protein. And then hopefully I can get some information from the structure and the sequence, and then maybe down the road publish a paper. So I've really learned, this has been a really, although I haven't made any breakthrough discoveries, I haven't published yet, it's been a really great project for me. It's exposed me what it's like to work in a lab. It's exposed me to the process of science. It's shown me that if you don't enjoy doing science for science's sake, you're gonna have a problem. If you're just waiting for results, you're gonna be miserable every day you have a failure. So it's been a really great opportunity for me. And if I don't get something by the time I leave Ohio State, the wonderful thing is that maybe the next person who comes after me will be able to finish up the project. So science, you can build on it like that. And that's really remarkable.
Andrew,
Mm-hmm.
How old were you when you started this research?
When I started this research, I was a freshman.
Age?
So I was probably 18, 19.
How old are you now?
21. So it's been 3 years, and I still haven't published. But I mean, I was only an undergraduate in college. It's not expected that you'll get something out of the way. The undergraduate experience is there for me to learn from. So, I learned that it was okay to make mistakes. I learned that it's okay to make mistakes as long as you learn from them. And that you'll make the same mistake however many times it takes you to learn from it. So, it's been a really, really interesting process.
Now, not many 18-year-olds would feel comfortable walking into a research opportunity of this type. What prepares you for being able to step into that arena?
Oh yeah, and I mean, I might not have been completely ready for it either. Well, I mean, I was ready, but I mean, I didn't walk into the lab and automatically know what I was doing. It was a learning process. But as I mentioned earlier, the internship class here at TVMSC really helped me out. Because by the time I got to college, I'd already been in at least 2 labs. So I'd been in Dr. Roan's lab at BSU, and then Dr. Jennifer, I forget her last name, Dr. Jennifer's lab at NNU. So I'd already been exposed to the laboratory setting. I already knew how to use a pipetter, at least I knew how to do that. I knew how to wash dishes already. I knew what luria broth was. So I came in with a good basic understanding of how a lab worked. And that allowed me to really kind of get some of the deeper details while I was in high school. So now I'm in a really good spot for graduate school. So since I have three and a half years of research experience, people keep telling me, that's really good. You can go wherever you want for graduate school. I'm still in the process of applying, so I don't know if that's going to be the case. I wish, but we'll see. But I mean, I'm really excited for it. At this point, although I haven't made any, breakthrough discoveries, I still know that science is what I want to pursue as a career. It's going to be something that's going to it's going to be the reason for me getting out of bed in the morning. And the reason for that is it's kind of, it wasn't that I woke up one day and I was like, you know what, science is what I want to do. It started off, I was just really good at it. I came here at TVMSC. I was good at math, I was good at science. And I thought it was interesting, sure, but was I like completely enthralled with it? No. That took time. It really required me kind of getting to know myself, kind of getting to know the world I was in to really understand that science was what I wanted to do. And if I can kind of put it this way, what really fascinates me about science is it discovers how the way things are. You know, it discovers the truth of the matter. So one of the things that really fascinates me is, so this is an amino acid, right? So amino acids are made of, you know, a handful of elements, handful of atoms: carbon, nitrogen, oxygen, hydrogen. But they arrange themselves in such a way, and they only arrange themselves in just a little over 20 varieties. I don't remember the exact number, which is probably bad. But they arrange themselves into amino acids. And these amino acids arrange themselves into proteins. And these proteins each perform a function for the cell. And the cell can only function when all its proteins work together and are in harmony with one another. So then these cells in a multicellular organism also have their own specific function. So, you know, my cardiac cells pump blood throughout my body. My brain cells kind of are able to take in all the information of my surroundings, process it, and then, you know, do something with it. My bone cells, you know, provide structure and support. When my bone marrow goes, every cell has a function. And everything works together to form a human body. And it doesn't stop there, right? So just like every cell in the body has a function, every human being here on Earth also has a function as well. Although we're not serving, we are serving a greater whole. We're serving society. We're serving civilization. So what we do is we, you know, as a society, we take the resources the environment has given us, and we use them to give each of our members of society life. You know, we use agriculture to take, you know, seeds, light, soil, and transform that into food to feed 7 billion people. I mean, we don't feed 7 billion people, but we could. You know, it's possible. Through the life sciences, we were able to combat plague and disease. With physics, we can fly through the air in an airplane. We can go to the moon. All of this stuff is made possible through science. And it's remarkable because never in a million years would nature come up with this on its own. It's only through logic and reason and intellect that civilization is able to uncover these mysteries and apply them to use. So that's just the really neat thing about the world that kind of motivates me to do science afterwards. But I mean, it took a while before I came to that conclusion, and my presentation can't do it justice. So what I recommend for you guys, the way is to discover what motivates you, discover what makes you happy, discover what you want to do in the world. And the way you can do that, the best way I found to do that is just to get involved. So colleges offer so many different clubs and opportunities. I mean, they've got, at Ohio State, we have hundreds and hundreds of clubs, probably well over 1,000. And I mean, you can find anything you want to do. So there's Quidditch League, Ukulele Club, cosplay, Amnesty International, Students for Free Thought. I mean, take your pick. And the purpose of these clubs isn't to set your life in stone. It's to figure out who you are. So, you know, join Happy Hour Club, you know? And if you discover that's your thing, well then great. But I mean, if after a few months of, you know, doing bar crawls, you're like, oh, this is killing my liver, I'm not feeling it. Well, then you know that's not you. So, you know, go ahead and join Biochemistry Club. And you're like, oh, hey, you know, biochemistry, all these people are really cool. They really enjoy biochemistry just like me. Maybe this is who I am. And I don't know, after, you know, a month, you're like, man, proteins suck. I don't want to do this forever. Well, then join Amateur Radio Club, you know? So the idea is that with every club you join, you know, you'll find a little something out about the club, but you'll also find something out about yourself. So that by the end of it, you'll have a better idea of who you are. Because as far as I understand it, and I mean, I've only been here 21 years, so I mean, by no means do I have full grasp of the matter, but life isn't quite about discovering the cure for cancer, or discovering the cure for AIDS. It's more about discovering who you are. And college is a wonderful opportunity for you to begin that journey and begin that process of discovery. So I'd like to just kind of finish with some words of wisdom that somebody told me. They told me, if you don't like who you are, what you're doing, who you're with, what you see around you, then change, then do something else. And that's the wonderful thing about being human, is we can do whatever we want. If you don't like where you're at right now, do something different. So, I don't know, that's kind of what I've learned in the past 4 years at college. I'm excited to go on to graduate school. I'm excited for you guys to go on to college for yourselves. I mean, you guys are gearing up for that. And like I said at the beginning, you guys are in a good spot. You guys are in a good position to go to any college you want to go to. A place like TVMSC is really going to open a lot of doors for you. But